infection, the various types of COVID vaccines, the salient features of some common vaccines, their preparation, their mechanism of action, then vaccination in special groups, and what is critical coverage of vaccines which would be required for eradication of COVID. So as we all know, this pandemic engulfed the entire globe in less than 3 months, from January 2020 to March 2020. Initially, there were only around 3 countries which started off in January, and by the end of March, there were nearly more than 180 countries in which the pandemic had spread. So how does a disease spread? The important thing epidemiologically is what we refer to as the basic reproduction number, or the R0 or R0 as it is referred. This is the expected number of cases which are directly generated by one case in a population where all individuals are susceptible to the infection. So in short, if there is one infected case, how many more infected cases that person can generate or create is the basic reproduction number of the disease. This is determined by the duration of infectivity of the affected person, the infectivity of the organism, and the number of susceptible people that the infected people come in contact with. As far as COVID is concerned, the basic reproduction number for COVID has been determined in many studies to be 3 to 8. When it is low also, there are studies which are saying that it may be, say, up to 1.5 in some areas. So a range of 1.5 to 8 is the basic reproduction number for COVID. So if we consider the R0 as 4, so in this cartoon we understand that if there is one patient infected, this individual further infects 4 more individuals, each of whom further infect 4 more. So if the R0 is more than 1, the infection can spread in a population, and if the R0 is less than 1, it is considered that the infection cannot spread in the population. So the strategy to control this spread primarily is to decrease the number of susceptible individuals. This is the community-based approach that can be used to control the spread of a disease. And how we do this? We do this by increasing the immunity of these individuals, and a major weapon for that we have is vaccines. But before we understand vaccines, we need to understand what this coronavirus is. So the SARS-CoV-2 virus, its structure primarily is that it has an envelope and a membrane. Inside that is the RNA viral genome, and on the membrane are various structural proteins, the important one being the spike glycoprotein. This spike glycoprotein consists of 2 subunits, the S1 subunit and the S2 subunit. The S2 subunit is the part of this glycoprotein that is attached to the envelope, and the S1 subunit is what has the RBD, the receptor binding domains, and it is in this S1 subunit of the spike glycoprotein that the receptor binding domains are present, which bind to the ACE2 receptors present on our cells or human cells. Now, as far as the genome is concerned, this is an RNA genome, and on this genome are 2 areas. the ORF1A and the ORF1B. In the ORF1B, from the 21,563rd nucleotide from the 5' end is the region that encodes for the spike protein. This region in the genome has been of major importance in developing vaccines, because this is the region that develops the spike protein. Now we also need to understand what this COVID virus or the coronavirus actually does inside our body. So when this virus enters or is exposed to human cells, it binds to the ACE2 receptors present on our cells, the host cells. And when this virus binds to these ACE2 receptors, it is taken inside the cell. Inside the cell, the genome of the virus gets duplicated using the protein synthesis machinery of the host cells, by which structural and non-structural proteins are made of the virus. The RNA is replicated, and that results in formation of multiple more viruses. And these viruses are then released from the host cell into the host circulation. Now here again, these virus cells are then picked up by antigen presenting cells. These viruses are picked up by the antigen presenting cells. These antigen presenting cells again bind to the virus via the ACE2 receptor, take this virus inside, and then they break up this virus proteins into small fragments, which are then represented or shown in the class 2 MHC molecules on these antigen presenting cells. Then these antigen presenting cells reach the secondary lymphoid organs like the local lymph nodes and the spleen. In these lymphoid organs, helper T cells, which are able to scan these antigen presenting cells, identify them and bind to these MHC molecules, and these helper cells then get activated. The activated helper cell converts some helper cells into cytotoxic cells, which release cytokines, and these helper cells also activate some B cells. The B cells that are activated in the local lymph nodes then undergo what is called as the germinal center reaction. These B cells in the germinal centers lead to the formation of plasma cells, which would then release these antigen specific antibodies, and these activated B cells in the germinal centers also differentiate into memory cells. I'm extremely sorry, there was some connection issue. Am I audible again, and is my screen visible? Extremely sorry. Uh, so I was discussing that the T cells which are activated lead to activation of B cells, and these B cells undergo germinal center reaction in the lymph nodes. These B cells, some of them differentiate. The B cells, some of them differentiate into plasma cells, which in the circulation would lead to formation of antibodies. The B cells also differentiate into memory cells. Now, these memory cells, when are released in the circulation, they identify some survival niches in the bone marrow where they remain dormant. And whenever the body would be exposed to this virus again, these memory B cells would immediately be able to respond in the form of formation of antibodies. Okay. In the germinal center, we also have what is referred to as affinity maturation, whereby these a better and more fitter antibodies are processed and prepared. So this is what is normally happening when the body is getting infected by the coronavirus. What we want to do is, by a vaccine, we want steps from here to be continued. That is, we want the vaccine to be able to be picked up by antigen presenting cells, which would present antigens of the virus, which would lead to formation of antibodies and memory B cells against those specific antigens. At the same time, what we don't want is the infection or the replication which would go about in our body. I will discuss this further in detail. So the SARS virus, SARS-CoV-2 or the coronavirus, basically has genetic material and some surface proteins. This genetic material is responsible for protein synthesis. The proteins again may be surface or non-surface proteins. This would then lead to replication, and replication causes disease, which determines the infectivity or the virulence of the organism. The surface proteins are involved in cell binding. They bind with the antigen presenting cells and lead to immunogenicity. So as a vaccine, what we want is something that has high immunogenicity and very low or minimal infectivity. This has been done multiple times in history. In 1880, it was done first by a method that is attenuated pathogens. We attenuate the pathogens to form vaccines. What was done in this was that the wild virus type undergoes repeated cultures in some foreign host, and in order to survive and adapt to this new host, the virus undergoes some mutations. As a result of these mutations, the virus's virulence decreases in human hosts, and then that attenuated virus is used as a vaccine. The limitations of this method are that it may take years to develop attenuated strains. And if we do not spend that much time in it, if we pick up the strains early which are poorly attenuated, those strains may rapidly revert to the initial wild type of genotype, and then inside the body can result in disease. Also, there may be some pre-existing cross-reactive immunity from natural contact with other coronaviruses. Also, attenuated vaccines cannot be used in immunocompromised individuals, as they may survive in some niches in bone marrow, and the virus, the attenuated virus, may revert to its wild type and then lead to severe disease. So because of the time-based limitation, no COVID vaccine candidate is using this strategy, has initiated clinical trials, because it has only been around 1.5 or 2 years since the COVID disease started. So no virus has been attenuated to an extent that it may be used in vaccines. The next way is by using what we call as the inactivated pathogen vaccines. In this, the virus is killed. In the previous method, the virus was only attenuated. It was still alive. It had just undergone some mutations. Here we kill the virus. This inactivation may be done either by heat or by gamma radiations or by chemical treatment by using chemicals like formalin or beta propiolactone. So, since the virus is inactivated or dead, these vaccines have a better safety profile. They are easier to transport and store because we don't have to worry about the viability of an alive or an attenuated virus. So one vaccine using this method, the inactivation method, is the CoronaVac, which has been developed by the Sinovac company, Chinese company, and they use beta propiolactone to inactivate the virus. Now this beta propiolactone, it binds to the guanine in the DNA chain, and it leads to, or rather the RNA, RNA of the in the DNA chain, and it leads to the formation of alkylated guanine. Now this alkylated guanine leads to opening of the guanine ring, abnormal base pairing, and excision. Oh, I'm sorry, there was some message on the platform. Yeah. So this alkylated guanine then inactivates the viral genome. So this inactivated virus particle added, and some adjuvants are added to this, and this, when injected inside the body, since it is inactivated, there would be no replication, but since it has surface proteins, these surface proteins would be presented to antigen presenting cells, and those antigen presenting cells would then present the surface proteins to B cells, leading to the formation of an immune response and immune memory. This vaccine has been approved for more than 18-year-olds. Its dose is 0.5 ml intramuscular in the deltoid region, 2 doses, 2 to 4 weeks apart. Its preparation is a refrigerated liquid containing a 40-dose vial. It can be stored at 2 to 8 degrees Celsius, but has to be protected from light. And its efficacy has been found to be 51 to 67% after 14 days of the second dose. The next vaccine using the same mechanism or a similar mechanism is the BBIBP-CoRB, also called as the Cobilo vaccine, which has been developed by the Sinopharm, again a Chinese company, and the Beijing Institute of Biopharmaceuticals. The mechanism here is the same. It uses an aluminum hydroxide adjuvant, inactivated whole virus, again approved for more than 18-year-olds. The dose is 0.5 ml intramuscular in the deltoid region, 2 doses 3 to 4 weeks apart. The preparation here is a single dose vial or a pre-filled syringe. It can also be stored at 2 to 8 degrees, has to be protected from light, and its efficacy has been found in studies to be 79% after 14 days of the second dose. The same mechanism has been used in the BBV152 vaccine, which we all know as the co-vaccine, which has been produced by Bharat Biotech in collaboration with the Indian Council of Medical Research and the National Institute of Virology at Pune. This is also a virus inactivated by beta propiolactone. An adjuvant of aluminum hydroxide is used. It has been approved for more than 18-year-olds. Its dose is 0.5 ml intramuscular in the deltoid region, 2 doses 4 weeks apart, and it can be stored at 2 to 8 degrees Celsius. Its efficacy has been found to be 78% after 14 days of the second dose. The limitations of these vaccines are that since these are inactivated, the virus is inactivated, it leads to a loss in immunogenicity also. So the vaccines may fail to induce a cellular adaptive response. As a result, we may need an adjuvant which stimulates the immune cells and amplifies the immune response. And also, these may need repeat and booster doses. So as we saw in the 3 examples of the vaccines, they all are using adjuvants and they all have 2 dose schedules, and boosters may be required in future. Next, we come to the subunit vaccines. Here, only a part of the vaccine is used. Which part? The part which generates a strong immune response. That part is identified, that immunogenic fragment is identified from the virus, and only that part is used as a vaccine. So these can be a protein subunit vaccine and virus-like particle vaccines. So in the protein subunit vaccine, the advantages are that these protein antigens, they are able to elicit the germinal center reactions which lead to affinity maturation of the antibodies, the isotype switching of the antibodies. So how do we synthesize the vaccines for these protein subunit vaccines? We either do a recombinant synthesis of the protein antigen, or the proteins are isolated and purified after a culture of the virus. Here, because only a part of the virus is used in the vaccine, it cannot lead to the disease. We use only the immunogenic fragment, and just by inserting the fragment, there is no way that we can develop the disease. So which is the most immunogenic fragment in the SARS-CoV-2 virus? Now that is the spike protein. And the spike protein also has multiple conformations, and it has been identified that the pre-fusion conformation that has 3 closed receptor binding domains is the most stable and most immunogenic conformation of the spike protein in the coronavirus. So the first vaccine that uses this strategy is the NVX-CoV2373, also called as the Covovax, which has been produced by the Novavax company, which is an American company. Now, this is a very interesting way of developing a vaccine. Let us see here. They identified the part of the COVID genome that is responsible for the formation of the spike protein. Now, this spike protein gene, they inserted into a baculovirus, which is a virus of insects. So this baculovirus, which had the gene for the formation of the spike protein, was then it infected a culture of moth cells. Moth, the insect, moth cells is where this baculovirus was injected, and it led to an infection. Now inside these moth cells, the baculovirus released the COVID genome, and the spike gene was then formed. So this, the spike protein, and this spike protein is then, it presents or expresses on the surface of these moth cells, and from the surface of these moth cells, these spike proteins are harvested. They are assembled around a PS80 or a polysaccharide 80 core. So the vaccine is this core which is surrounded by these recombinant spike proteins that we formed in conjugation with a matrix M adjuvant. And this is a plant-based or a saponin-based adjuvant. So because it is plant-based, it leads to a very robust immune response. So this matrix M adjuvant has been added to this protein subunit vaccine. This vaccine is being used in more than 18-year-olds. Its dose is intramuscular deltoid, 2 doses 3 weeks apart. Its preparation is a 10-dose vial stored at 2 to 8 degrees Celsius. Phase 3 trials only are going on. This has not yet been approved. And in phase 3 trials, it has shown an efficacy of 89.7%. The limitations for these protein subunit vaccines are that there is a very small size of the antigen, which diminishes the uptake by the antigen presenting cells. The antigen presenting cells do not pick up very small proteins. They are good at picking up substances which are of the size of viruses or other bacteria. But very small sized antigens are not picked up nicely by the antigen presenting cells. And also these protein subunit vaccines are less immunogenic, so they require an addition of an adjuvant, and they may require more booster doses in order to have a stronger and more durable immunization. The next subunit vaccines are the virus-like particle vaccines. Here, empty virus particles are used. These particles have no genetic material, so again, they cannot be infective. They are extremely safe. And these particles have several copies of the same surface antigen. So these mimic virus structures and as a result have a strong immune response. A vaccine that is using this method is being produced by Medicago, which is a Canada-based company, and GlaxoSmithKline. The vaccine is called CoVLP, and it is an extremely interesting method that they are using. Here, again, they identified the spike genome. Then this genetic material was made into a liquid suspension, liquid containing this genetic material, the spike gene. Into that liquid, they introduced plants. The plant that they are using is the tobacco plant. So they introduce this tobacco plant and then expose it to extreme vacuum. In the presence of the vacuum, the leaves of the plant pick up this genetic material. Then these plants are incubated, say for around 2 weeks. And while they are incubated, these plants act as ways of expression of this genetic material. The genetic material inside the leaves is expressed and virus-like particles are formed inside the leaves of the plant. Then these leaves are harvested, just broken from the plant, and from that purified virus-like particles are isolated. Well, these are micrographs. This is the real coronavirus, the SARS-CoV-2 virus. And this is a micrograph of the Medicago virus-like protein that has been, a virus-like particle that has been formed inside the leaves of the plants. This uses an adjuvant which is called the AS03. Its dose is intramuscular deltoid, 2 doses 3 weeks apart. Again, it has not yet been approved, only phase 3 trials are ongoing, but again, very promising. Next, we come to a way of creating vaccines called as the viral vectored vaccines. Here, we are using some other virus which acts as a vector to transmit the genetic material of the coronavirus. Again, we only transmit the genetic material responsible for formation of the spike protein. So this vector virus, when contains the genome for the spike protein of coronavirus, again cannot lead to coronavirus disease. So adenovirus, measles virus, or vesicular stomatitis virus, these are used, they are modified, which results in reduced virulence of these viruses, reduced replication potential of these viruses, but the ability to infect cells is maintained. This leads to delivery of the pathogen's genetic information to our immune cells, which express and present the antigen proteins to the lymphocytes. Now, these viral vector vaccines can be of two types. The vectors can be of two types. These may be either replication competent or replication defective. By replication competent, we mean that the vector is competent to replicate, it multiplies. So a lower dose of the vaccine is required, but the disease that the vector causes, the individual may be susceptible to that. In replication defective, the vector does not self-propagate. So even though a higher dose may be required, but these are extremely safe and we will not even have the disease that the vector may cause. The few limitations for these are that we may have some pre-existing immunity against the vector virus. And since if we use the replication defective vectors, then some booster doses may be required. A primary and then followed by a booster regimen may be needed. And in these primary and booster regimens, it has been found that there is decreased immunogenicity of the boosters. That is, the level of immune response that we get after a booster is extremely reduced. The cause for that is that the body forms antibodies against the viral vector after the first immunization. So in the second immunization, when we give the shot, the body already has antibodies against that vector and it does not let the vaccine act. So the solution for this, for the fact that there may be some pre-existing immunity, is that we use some less common vectors. So if we use some less common vectors, there are lesser chances that individuals would have pre-existing immunity to those. And we may also use non-human viral vectors. So if there are some non-human viral vectors, obviously we would have, there is no chance that we would have a pre-existing immunity for them. And so such vaccines may act better. The first in this type is the Jensen vaccine, which has been produced by the Johnson & Johnson and the Beth Israel Diaconess Medical Center at Boston, also referred to as the AD26-CoV2S vaccine. In this, a replication defective human adenovirus serotype 26 is used as a vector. So inside this adenovirus, we insert the coronavirus gene for spike protein, and this is then injected into the individual. This virus is picked up by the antigen presenting cells, which form antibodies against the spike protein that is present inside the genome of this virus, and our bodies are able to form antibodies and protect us from any virus which would contain those spike proteins. So they have used the rare serotype of adenovirus, that is adenovirus 26, and so fewer people would be exposed to this, so there would be lesser pre-existing antibodies. It is approved for more than 18-year-olds at a dose of 0.5 ml IM deltoid, single dose, and a rare side effect that is seen, say 3.5 in 10 lakh people, is thrombosis with thrombocytopenia syndrome, and this is seen 3 to 15 days after vaccination. This is again it has to be stored at -20°C. It can be stored for 2 years at 2 to 8°C only for 3 months. Within those 2 years, it has to be protected from light and has to be discarded within 6 hours of opening. The efficacy of this vaccine is 66.9% after 14 days and 85.4% after 28 days. The other vaccines that are using this is the ChAd-OX1 recombinant vaccines. The one that has been produced in collaboration with the Oxford University. In Oxford University, in collaboration with AstraZeneca, is making this, named as the AZD1222 Vaxzevria. In collaboration with the Serum Institute of India, it is called the Covishield, and in collaboration with the SK Bioscience company, it is called as the SK Bioscience Vaccine. Here, the chimpanzee adenovirus is used, so a non-human adenovirus is used. It is modified to be unable to cause disease, and in this vector, again, the genes for the spike protein are inserted, and then this vector is the vaccine. This vector in combination with this gene become the vaccine, and this, when injected, this leads to protection against the COVID disease. This is also recommended for more than 18-year-olds. An IM deltoid injection of 0.5 ml, 2 doses, 4 to 12 weeks apart, which in India has been, by the government of India, has been approved only 12 weeks apart. Side effect again seen in 4 in 1 in 10 lakh people is thrombosis with thrombocytopenia syndrome, seen 4 to 20 days after the vaccine. And this again, vaccine has to be stored at 2 to 8°C. Its efficacy has been found to be 63 to 76%. The next is the GAM-COVID vac, or the Sputnik V vaccine, produced by the Gamaleya National Center of Epidemiology and Microbiology in Russia and the Russian Direct Investment Fund. And in India, there is a collaboration with Dr. Reddy's. Now, the limitation of the vector vaccines that the body may develop some immune response after the first dose has been countered in this vaccine, because this vaccine uses 2 different adenovirus vectors for the first and the second dose. In the first dose, it uses the vector AD serotype 26, and in the second dose, it uses AD serotype 5. So as a result, even if the body forms antibodies against adenovirus serotype 26, when the second injection is given, the vector has changed. It has become adenovirus serotype 5, and therefore those pre-formed antibodies do not affect the response of the vaccine. So it has been found that after the second dose, the Sputnik vaccine has a very high immune response as compared to other vaccines which are using the same vector. This is again approved for more than 18-year-olds. There are two doses, 21 days apart. It's a 0.5 ml IM dose. The preparation here is that it is frozen at minus 18 degrees Celsius, or there is a lyophilized preparation that can be stored at 2 to 8 degrees Celsius. The efficacy of this vaccine has been found to be 91.6%. Now another vaccine is the Sputnik Light. Sputnik Light is nothing but the first component of the Sputnik V vaccine. So if we get only the first component of the Sputnik V vaccine, single dose IM, that has a adenovirus 26 as the vector, then that is the Sputnik Light vaccine. This again has been found to have an efficacy of 79.4% after 28 days, and this has also been approved in Russia. Now next are what we call as the nucleic acid vaccines. These are again fast to develop. They consist of the genetic sequence that encodes for the viral antigen, and they also have a delivery platform with it. They are again potent inducers of the immune response. There are two types of nucleic acid vaccines. They may be either DNA vaccines or mRNA vaccines. The DNA vaccines can be used via multiple routes. There are trials going on and vaccines are being made that may be used by the intradermal, subcutaneous, transdermal, IV, IM, nebulization, or the oral mode. In all these, the host cells, antigen presenting cells, would pick up this DNA, bind to it, form spike proteins inside them, which would then lead to an immune response. So the advantages are that they are quite stable and can be stored at plus 4 degrees Celsius. So their distribution becomes extremely simple. An example of this DNA vaccine is the INO 4800, which is being made by the Inovio and the International Vaccine Institute in the USA. Here they have again selected the spike protein gene, formed this DNA plasmid. This DNA plasmid is a small electric current which leads to picking up of this DNA inside the host cells. And then when this operated in host cell, the immune response. 2 and 3 trials are ongoing. The next is the Zykov D Vaccine, formed by Zydus Cadila in collaboration with the Department of Biotechnology, Government of India, and the Biotechnology Industrial Research Assistance Council. This is again a DNA plasmid vaccine. The vaccine is intradermal, and the important thing here is that it is a needle-free, spring-powered jet injector. There is no needle for this vaccine. They have a separate injector, which by a spring action, the thermal maybe 3 doses, dose analysis in 6.6%. The Zydus Cadila have applied to the Drug Controller General of India and is seeking emergency use authorization for this vaccine. Next, we come to mRNA vaccines. Here, these vaccines have a lipid nanoparticle capsule, which helps the vaccine or the particles inside it to penetrate through the cell membrane. Now, once penetrated, it delivers the mRNA into the cytoplasm. And this mRNA, in the presence of the cytoplasmic ER ribosomes, leads to the formation of the foreign antigen proteins, and which are, when presented on the surface of the cells, lead to an induction of the immune response. The limitation of the mRNA vaccines is that they are extremely unstable. A minus 70 degrees to a minus 20 degrees Celsius temperature has to be maintained for their long-term storage. And as a result, they have a complicated distribution logistic. The solution is to keep them for 2 to 8 degrees Celsius for 6 months. We have to add either specific mutations in the mRNA or some stabilizing chemical modifications might have to be done to the mRNA. So a common vaccine using this principle is the mRNA 1273 vaccine, also called as the Mrna vaccine, which is being produced by the company Moderna in collaboration with the National Institute of Allergy and Infectious Diseases of the USA. Here, the outer capsule merges with the host cell and delivers the mRNA. This mRNA uses the ribosome in the cytoplasm, leading to the formation of the protein. These spike protein then are expressed on the surface of the cell and thus lead to an immune response. This vaccine is approved for more than 18 years of age. Its dose is 0.5 ml intramuscular, 2 doses 28 days apart. A 10-dose vial is available. Its preparation is a frozen suspension at minus 25 to 15 degrees Celsius. It has to be thawed before use. After thawing, 2 to 8, it can be stored at 2 to 8 degrees Celsius for up to 31 days. However, once the vial is used, it may be kept at 2 to 25 degrees Celsius, but has to be discarded within 6 hours. The efficacy of this vaccine is 94.1% 2 weeks after the second dose. Similar mechanism is used in the mRNA BNT162b2, also called as the Comirnaty, or the Pfizer vaccine produced by the company Pfizer in collaboration with BioNTech, a German company. Again, similar mechanism. It has been approved for more than 12 years of age. Its dose is 0.3 ml intramuscular, 2 doses 21 to 28 days apart. This is also available only in multi-dose vials. It is available as a frozen suspension, has to be thawed before use. Its efficacy has been found to be 95% 1 week after the second dose. But again, cold chain maintenance continues to be a major drawback of these vaccines. So to summarize, the COVID vaccines may be either inactivated virus like the CoronaVac, BBIBP, or the Covaxin. Subunit vaccines, the protein subunits as in Covovax, the virus-like particles in CoVLP. Some viral vector vaccines like the Janssen, Oxford, or the Sputnik vaccines. The DNA vaccines like the INO and the Zykov D, and the mRNA vaccines like the Moderna and the Comirnaty. So out of these, the approved vaccines, the WHO approved vaccines are the Moderna, the Pfizer, the Oxford, the Janssen, Sinopharm, and the Sinovac. As the Government of India approvals are concerned, the Government of India has approved the Covaxin, the Covishield, which is the Oxford vaccine, and the Sputnik V vaccine. Vaccination in special groups in pregnant women, it has been approved because the benefits are more than the risk. Similarly, in lactating women also, vaccination has been approved. For older individuals, there is no upper age cutoff. For adolescents and children, trials are going on. Only the Pfizer vaccine that can be used for more than 12-year-olds. Apart from that, all other vaccines can be used only for more than 18-year-olds. In immunocompromised individuals also, vaccination is approved because none of these vaccines uses a live virus, so they cannot cause the disease. However, they may have a suboptimal response in immunocompromised individuals. So let us quickly see what is vaccine efficacy and effectiveness. It is the ability of the vaccine or the reduction in risk of getting the disease as a result of taking the vaccine. Efficacy is when vaccinated individuals are compared with unvaccinated participants in clinical trials, and effectiveness is what we see in real world studies. So if it is a trial, the result of a trial gives us the efficacy of the vaccine, and the real world studies give us the effectiveness of the vaccine. So what is critical coverage? Our long-term goal is the eradication of the disease. So critical vaccination coverage means the minimum proportion of the population that must be immunized. This is the concept that is used in what is being referred to as herd immunity. So the critical coverage, there is an epidemiological formula which is 1 minus 1 upon R naught divided by the vaccine effectiveness. So if the R naught here in this table, R naught is represented here, the possible vaccine efficacy is represented here, and what these cells give us is the required critical coverage. So if the R naught is extremely high, we need near 100% vaccine coverage in vaccines which have an efficacy of up to 80%. And even if the vaccine has an efficacy of 99%, if the R naught is 8, that is, the disease has very high potential to spread. 88.4% of the population would need to be immunized. So even if the R naught of the COVID virus may be 3, for a R naught of 3, we need 95% coverage of vaccines which are 70% effective, and near 70% coverage of vaccines even if they are 100% effective. So at least 70% coverage would be required if the R naught stays to be something near 3. So what I want to emphasize here is that only the vaccine cannot be our whole and sole way of controlling the disease. We need to continue to have precautionary measures, because expecting that the vaccine coverage is so high in such a large population in India would be something not very near. We need to continue our COVID appropriate behavior in order to win this fight. Thank you so much. I am open for any questions that you may have. should I close the class then? I will end the class. Thank you so much, everyone. It was a pleasure. Thank you.